So the beginning of translation starts with initiation. So we have initiation, then we have elongation and termination. Initiation is going to start at the start codon, which is, again, methionine or AUG. So when this happens, you have the ribosomal subunit come together on the messenger RNA and it allows the transfer RNA that carries the anticodon. So if AUG is the codon, UAC is the anticodon, and it carries also with it the methionine. Oh, it always does this, doesn't it? There we go. So once that happens, then the next transfer RNA can move in and bind. So the first transfer RNA moves in to a region called the P site. The second transfer RNA will move into the region called the A site on the ribosome. These two amino acids that are linked to the transfer RNAs are going to unbind from the transfer RNAs, and they're going to form a peptide bond between the two. And then the entire ribosome is going to shift. When the ribosome shifts, it moves the first ribosome into a site known as the E site, or exit site. The second ribosome, or the second transfer RNA that was there, is going to be now found at the P site. And now a third transfer RNA can move in. And so let's look at what that looks like. Here you have the first transfer RNA with the anticodon and the appropriate amino acid, methionine. Here's the large and small subunit. They're going to come together. Once they come together, now the transfer RNA can bind to that first site. Once that happens, a second transfer RNA is going to move in and bind to that second site. And then the entire ribosome will shift over, and the ribosome that was in this spot, the P site, moves to this spot because the whole thing moves, but the transfer RNAs don't move. They just stay because they're bound. And once they hit that this spot here, then that's the exit site. The transfer RNAs are going to unbind and they're going to leave, but they leave behind that amino acid, which binds to whatever is in the second site here, which is called the A site. So we have the P site, the A site, and then over here is that E site. And this is going to continue until we hit the release factor, or that stop code, and that's the termination. So termination occurs when we hit that codon that does not code for an amino acid. And once that happens, then the polypeptide is going to be released and is going to be able to fold into its functional form. Sometimes this is going to require extra modifications to the protein. Maybe iron's going to be bound, maybe we're going to have carbohydrates bound, whatever it is. And that's going to make that protein now more functional protein. And the ribosome can come apart, but the ribosome can produce, so ribosomes aren't used up, they can be reused to make more proteins. The transfer RNAs are not used up. They're going to move out and they're going to pick up new amino acids. So they're ready for the next protein synthesis. The transfer RNA can be retranslated. The messenger RNA can be retranslated if it's not been broken up. If it's broken apart, then of course it's not going to be retranslated. So the process of transcription and translation can be coupled, which means you can be transcribing and translating at the same time in prokaryotic organisms. This is because both transcription and translation occur in the cytoplasm, and the transcript that you're producing for prokaryotes codes only for proteins. So the initial transcript is what we're going to translate. In eukaryotes, they are not coupled, and the reason for that is twofold. So the first reason is because we transcribe in the nucleus, but the other reason is because the transcript is not going to be considered mature. We first have to modify it. So remember from your Gen Bio classes or from Anatomy and Physiology when we talked about the modifications that have to occur before protein synthesis can occur? You first have to take out all of the regions of DNA that don't, or all of the regions of the messenger RNA that don't code for proteins. And so that's a huge amount of the material. So I like to ask my class, and I'll ask you as well, how much of our DNA do you think actually codes for proteins? And so you can answer it obviously to yourself. I'm not right there in the room, but it's something I may ask at some point during class time as well, or maybe on an exam. So it's something that you might want to look up, or if you can't figure it out, ask me, okay? But because there's a huge amount of our DNA that doesn't code for proteins, when we are transcribing that DNA, we are transcribing non-coding regions. Those non-coding regions are termed introns. And so we first have to take out the introns, and then we're going to splice together the regions that do code for proteins, and those are called exons, expressed portions. We do that, we add a guanine cap and a poly A tail. And once we have that all done, then we can let the messenger RNA move into the cytoplasm, or the rough endoplasmic reticulum, where it'll bind to the ribosomes and will have translation occur. So they are not coupled. And so here you have that DNA. We have the pre-messenger RNA. You can see the regions here that are introns that we're going to get rid of. Here we have that proper messenger RNA. We add the poly A tail, the guanine cap. And then that moves out into the cytoplasm where we're going to translate. Over here, while it's being transcribed, we're already translating it. So we've talked about protein synthesis. And proteins are what our DNA codes for. So if there's a mutation within a protein, that can cause changes in the organism. So a mutation is an inheritable change in our DNA. So it's something that we take and we can pass on to our offspring. This can change amino acid sequences, and it can alter the functionality of different proteins at times. There are lots of different types of mutations. So we have mutations called point mutations, which affect a single nucleotide. And when you have one single nucleotide that changes, that can cause major effects, but sometimes it doesn't do anything. And so a point mutation can be called a silent mutation if the codon that codes, that is being coded for, is the exact same amino acid, and I'll show you what that looks like in just a minute. It could be called a missense mutation, where it codes for a different amino acid. So one nucleotide changes and an entire different an entirely different amino acid is produced. Or it can be a nonsense mutation if the codon changes to a stop code. The other type of mutation is a frameshift mutation. So frameshift mutation occurs when you have the insertion or deletion of one or more nucleotides from the DNA or to the DNA. And this oftentimes changes the it doesn't oftentimes. It always changes the reading frame, and that change in reading frame typically will alter the amino acid sequence and oftentimes codes for a stop codon in the middle of the protein, so you end up having a non-functional product. So I'm going to stop here, and we'll get into these mutations in the next video. All right? Bye.